Can you see my slides now?
Yeah, we can see your slides.
Yes, you can start.
Okay.
Okay.
Hello and good morning to everybody. Good afternoon. Good time to everybody. Today I'm going to talk about the arrhythmogenic effects and tissue of the hydroxychloroquine and azithromycin in the treatment of COVID-19. I would like to mention that this work was done in collaboration with Ilya Uzalek from Georgia Tech, Dr. Shahriar Iravanian, Elizabeth M. Cherry, and Flavio Fenton. So to start the background, I think there is no need to mention about the ongoing pandemic and the toll that it is taking on the global society. And from the very beginning, hydroxychloroquine was introduced as one of the possible treatments for the COVID-19. And this drug is a slightly less toxic derivative of the chloroquine that is used to treat malaria and autoimmune conditions known to cause QT prolongation. Azithromycin, on the other hand, is used to treat a wide variety of bacterial infections. It works by stopping the growth of bacteria. It is also known to cause QT prolongation and sudden death by blocking the potassium channel and thus slowing the cardiac repolarization. So HCQ alone and in combination with azithromycin received significant attention, independent of the limited evidence in the efficacy of HCQ and azithromycin. So today we are going to talk about the important safety questions. Before COVID-19 pandemic, their combination was rarely used. So it is important to see how they would react when they are used together. And both of them are known to use, known to increase the QT interval. And we want to know if they are combined, what would be the effect. So while the HCQ was regularly used to treat malaria, the doses that was used for HCQ is much higher. So for the treatment of malaria, it is typically 400 milligrams of HCQ once per week. As for the treatment of COVID-19, 200 milligrams 3 times daily to 400 milligrams twice daily was used. And it is important to mention that HCQ accumulates in the body and has a washout period of around 2 weeks. So if we are using these large doses, it can cause, it can be serious, the effects of the HCQ. And since both of them are increasing the QT interval, we want to know what is the combined effect. On the other hand, the patients with COVID-19 developed left ventricular dysfunction, cardiogenic shock, and various cardiac arrhythmias. And having QT prolongation could be even more fatal for the patients. And another side effect of COVID-19 is it can cause hypokalemia, which increases the probability of arrhythmic events. And with HCQ and azithromycin combined, it can exacerbate the arrhythmic events even further. So the HCQ primarily blocks the funny current, the L-type calcium current, the rapid delay rectifier current IKR, and the IK1, and the sodium inward current. And azithromycin usually increases QT and affects the IKR channel and the L-type calcium channel and the sodium channel as well. So these are the major channels that are affected by the two of the drugs. The way that we are going to do the study is that because we have the capabilities to do both experiments and the simulations in the lab, the first thing that we tried to do was to do voltage optical mapping on the Langendorf perfused guinea pigs hearts. 12 of these animals were used for these studies, and then we tried to what we did next was to do numerical simulations in tissue for a guinea pig model, the Lifshitz Ruby or LRD model. And then we tried, what we did next was we started doing the simulations in tissue to extrapolate the results into a human model, namely OVVR model. So the first part was to quantify the effects of on the normal part with no drugs, and then HCQ alone and azithromycin alone, and 6 of the animals were used for the HCQ and azithromycin combination, and quantifying the spatial temporal variations. APD and CV restitutions across the left ventricles were calculated, as well as alternance and ECG for all cycle length of all 4 cases. And we use the normal experimental data to fit our calibrated GP model. And later we applied the drugs in the numerical model by applying the blockages to validate the experimental data, to validate against the experimental data, and then be extrapolated to the human model. So the reason that we are using the guinea pigs is that they share expressions of many ionic currents that are similar to the humans. The action potential may not be exactly similar, but the ionic currents that are involved in this type of a study, they share resemblance. So some of the results can be seen here, that the B refers for before and after introduction of the drugs, and APD 250 is for action potential durations recorded at 250 milliseconds pacing during the natural SA node pacing. And then we can see that for the control cases, the results don't change significantly before and after, but when the drugs are introduced, the before and after are significantly different. This has been done for the azithromycin and the HCQ combined. You can also see the conduction velocity before and after of introducing the drug. One interesting result was the alternance. So ACL refers to the pacing cycle length at the onset of the alternance. So you can see for before scenario, those columns that are empty, there was no alternance. But for the after scenario of the introduction of the drugs, you can see that for all the cases, we observe alternance. Also, you can see the amplitude of the alternance for those cases that the alternance was happening was increasing. The other part that was happening, you can see the cycle length, the minimum pacing cycle length increases for the onset of the alternance when the drugs were introduced. If we look at the restitution data, what we can see that the control doesn't change after 4 hours significantly, and there is still a good overlap between the restitution data, whereas for the introduction of HCQ only, the action potential durations increase significantly, as well as the slope of the restitution curve. For azithromycin, the action potential duration and the slope increases, but not as significantly as for the HCQ, and the combination seems to be somewhere in between the two. So some of these results are already published. Another part that you can see is the alternance. So the top left figure is the control set that you can see alternance, slight alternance happen at very small pacing cycle length, whereas for the combination of the two drugs, alternates start to appear much earlier. Also, the magnitude of the alternates is much larger. And also the minimum pacing cycle length drops, increases as compared to the control. Also, if you look at the ECGs, this beautiful data that was recorded during the experiments, these little blips here are the pacing stimulus. So first you see the pacing, and then the QRS complex and the T waves. So without the drugs, you can see that the QT interval is about 1 block, whereas when the combination of the drugs were introduced, the QT interval significantly increases. As well as after 4 hours of the drugs being perfused, you can see T-wave alternates as well in the results. So another result from the optical mapping that you can see here, without the introduction of the drugs, even at 140 milliseconds of pacing, the propagation of the waves are homogeneous, whereas you see with the introduction of the drug, even at 380 milliseconds, there are significant dispersions in the tissue. And here you can see, after 2 paces, some sort of arrhythmic event happens here that could be an ectopic beak or conduction block or re-entry, and we can't know for sure which one was it exactly due to the 3D effects that exist. So the numerical results are already good. Why should we, the experimental results were already good. Why should we bother with the numerical results? So the numerical results provide a pathway to extrapolate to the human data. And on the other side, they have been suggested to be used in drug trials by the CEPA initiative. So this initiative is sponsored by FDA, and its goal is to develop new paradigm for cardiac safety evaluations and drugs that provide a more accurate and comprehensive mechanistic-based assessment of arrhythmic potential. But the problem or the shortcoming regarding this is that the CP studies rely only on single cell studies and do not consider 1, 2, or 3D effects, whereas it has been shown in the literature that coupling is an effective nonlinear current that can dramatically change the cell's dynamic. A classic example is the Hundredly model that just due to the coupling, the sodium current is delayed, and that in turn turns into the delay of calcium release that overall increases the action potential by as much as 18 milliseconds just by pure coupling in the tissue. So the challenge right now is that we have to deal with stiff ODs. We have to deal with a large number of state variables. We have spatial discretization, small spatial discretization that requires 10 to the 10 ODEs per second of physical time to be modeled. So our solution is GPUs. So instead of supercomputers, we use GPUs, and there are various choices. And one of our previous studies, we had shown that WebGL outperforms the other implementations significantly, especially if the model is more and more complex. Moreover, it is interactive, it is machine independent, and it's free. So you can see you can even do full 3D simulations on a cell phone that is also interactive. One of the examples that you can see here is a reaction diffusion model that you can run it so fast that it's faster than real time, and you can still interact with it. Here it seems that the solution is stationary, whereas actually there is a stroboscopic effect going on, and this is a real-time simulation, not an animation. So what we do is an image manipulation problem in WebGL that we treat the entire solution as multiple images, and we assign physical values to the color channels, and we start manipulating those. So after you assign color channels to each of your physical variables, like for example, to the red channel of color 1, you can assign the edge gate and so on and so forth. The rest of the coding is very similar to a C language. So for our numerical setup, we use the 2D for both LRD and OVVR. The LRD gates were updated using adaptive time stepping, and central differences were used for spatial discretization, and convergence studies were done beforehand. But the LRD model, this was our control set, and this was the action potential, the APD resolution for the original LRD. So we had to calibrate the LRD model first, and then after we calibrated the LRD, we blocked the currents, and these were the results that we got. The red one is the azithromycin only. Then the green one is in between the HCQ and azithromycin, and then you can see the HCQ only, which still shows the action potential prolongations, APD prolongations for the same DIs, and shows the same trend where the results qualitatively match what we see in the experiments. Also, we see for the alternance, what we see in the experiments can be seen in the numerical results, whereas in the numerical results, the nature of the alternance that we see and the bifurcation that we see is different from the mechanistic bifurcation that happens for the experimental results, which is one of those documented cases for the numerical models. Also, on the conduction velocity, we can see the supernormal velocity also is observed in the numerical data, so in a similar fashion to what we see in the experimental data, but the trends could be different. Also for the combination of the drug and combination of the two drugs. So these are the observations. The supernormal conduction is seen in some of the recordings, but the one before or after may not show it. It seems that supernormal conduction occurs in a small region of the parameter space. And experimentally, supernormal conduction is seen at the time of APD and CV alternance. In modeling, it is only seen with HCQ in combination with azithromycin. Then we extended the simulations into the human model, which showed a similar trend, which, if you notice, the minimum diastolic interval increases significantly for the HCQ and azithromycin combinations when we are applying it to the epicardium cells for the OVER model. If you're interested to know more about the WebGL simulations, these are some resources. We have an ongoing course at Georgia Tech that we are frequently putting the video lectures online. You can visit this GitHub page for more information. Some of the lessons that we learned that HCQ and azithromycin both showed arrhythmogenic effects, but when they were combined, the effects were slightly less pronounced. The HCQ and azithromycin can lead to supernormal velocity in both numerical and experimental results. The onset of alternance shifted to higher pacing cycle length with the introduction of the drugs, both numerically and experimentally. And the results seem to hold as more profoundly as we extrapolate to human data. The numerical results are at most as accurate as the models that we use to solve. So we are at the mercy of the model, how good the model is, and then it determines our numerical simulation results. While qualitative agreement exists with the numerical results and the experiments, the quantitative agreement still can be improved. And another point is that while we can use the numerical results as a sign, as telltale signs, warning signs to avoid catastrophic clinical results, we believe they are not yet a substitute for animal and human trials. And model tools allow for in-depth analysis of cardiac dynamics, even on personal devices, in a timely manner. So please consider using tissue simulations rather than single-cell models. In the end, I want to thank the support from the NSF and our team and team members at Chaos Lab, and especially thank Flavio for his mentorship. And thank you for your attention.